Okay, thank you very much. So it's a great pleasure to see the younger generations again. And now I think we immediately will start with Sarah Hearne. She represents the international organizations for plant research that was started in around 1940 in CIMMYT with the Rockefeller Foundation, and the grow out to the what's called the CGIR. And now we will hear from her, who is in the principal investigator for the corn and wheat breeding, how the work is going for the finding in the varieties, the wild varieties, the genes that all plant breeders dream of. So please, Sarah.
Thank you, Mark. Okay, so I want to talk to you today about sustainability, and in particular, what the contribution is of the international public sector breeding, specifically that within the CGIAR. So the CG was formed as an entity in 1971, before many people here were born. It's a global partnership of 15 research centres across the globe that collaborate to advance the agricultural development agenda. Now, very recently, we formed a new entity, which is called 1CG, where 12 of these centres are coming together to combine under one umbrella with a new research agenda that really looks at broader partnership to drive agricultural development, particularly focusing on the Sustainable Development Goals. So the SDGs, so we're talking about sustainability. There are 17 of them. You can go onto the SDG website to look at the UN goals. In particular, the work of the CG focuses on larger effect things like by 2030, ending hunger, ensuring access by all people, in particular those in poor, vulnerable situations, access to food and other resources. So the CG works in 5 main areas of the globe, including Latin America, the Siwana region, Western Central Africa, East and Southern, South Asia, and the Southeast Asia and the Pacific. We work across 5 main impact areas, which are outlined here on the screen. And what I want to talk to you today about, in particular with the SDGs, is the impact that breeding can have towards poverty, hunger, and climate adaptation. So at the moment, we have around 690 million people globally. That's about 9% of the population who are hungry. This information came from the 2020 State of Food and Nutrition report from the FAO. Now here I'm giving you maize and wheat as an example. We could scale this across other species, other commodities, staples like roots, tubers, bananas, other legume crops. But essentially, we have an increasing demand for these commodities. This demand comes not only from population growth, but from changes in diet. And the projected demand is here. This is the demand projected for wheat and maize by the FAO by 2050, so a big increase in demand. If we take the current trends of productivity improvements, they're seen here in the dotted lines underneath, we're not going to meet the demand by 2050. The additional challenge that we have is we've got disease, we've got the impacts of climate variability, climate change, we've got an increasing competition for resources such as water, nutrients, energy in agriculture, which all act to push this curve down. And what we try to do within the CG is devise new tools, new methods, new approaches through agronomy and through breeding to try to push this curve back up again, to try and meet these needs and demands. So if we look at climate change specifically, and I just pull out two factors that we all hear commonly about, which is heat and drought. Here is some analysis from 2011, but from David Labelle's lab, where he took historical data from 20,000 yield trials that we'd run in maize across Africa. And extrapolating from the data from the yield trials, for every degree day above 30 degrees C, we get lower yield under both optimal conditions, conditions that we would all like to see farmers having, where you have sufficient fertilizer, you have very, very good soil husbandry methods, you've got enough water availability. Even there, we start to see a decline in yield into higher temperatures. And under drought conditions, that effect is even more acute. Now, when we look at the germplasm base that we have for breeding, many of the current donors for droughts and the key parental lines that we use in the development of new hybrids are susceptible to drought stress at elevated temperatures. So we start to get a challenge of combined effects of these two particular stresses, which are challenging yield and causing a lot of issues when we look at crop production in Africa and beyond Africa. So this isn't just a textbook example. This is what drought looks like in the field. These are examples coming from the BBC. We see this when we work with farmers ourselves in Zimbabwe and other regions. Drought impacts food security massively, food availability. You get added effects here. You start to get issues of disease coming into these ears, aflatoxin, other challenges, all piggybacking on the impacts of drought and heat stress. If we look at maize yield projections, I start this video. This is a recent analysis that came out earlier this month in Nature Food. There's been quite a lot of publications looking at climate change coming out recently on the back of COP26. And what you see here is a challenge pretty much everywhere in terms of maize yield projections as we move forward this century. Apologies to anybody who's red-green colorblind here. This is not my video, not my color scale. Those who are colorblind, most of the things are turning red, which is bad in this regard. So in many of the regions where we grow maize, either as a feed crop, which is predominantly in the US, and as a food crop, which is the predominant use in the global south where we tend to operate, we see a significant impact of climate change on maize yield. So just to give you a quick overview of what CG breeding looks like. The core of the breeding really focuses here on population improvement. And population improvement really is a closed population of elite by elite crosses where we drive hard and fast those populations to get as much transgressive segregation as possible. And from these populations, both in the CG and within our NARS and private sector partners, we start to extract materials that go through variety testing. Now, all of this is guided by farmers and consumers through the development of product profiles, which describe what variety should look like, and there are breeding targets. Now, within this, we will get lists of things, lists of ingredients that we need in terms of traits in product profiles that sometimes we don't have sufficient variation for within the existing elite pools that breeders are using. When this occurs, we start to look at donor and development, trait development. New donors that we can use to introduce new variation that's of value for specific breeding targets. Sometimes these donors exist in other germplasm pools, we can pull them in. Other times we have to go through trait mining. And what I want to focus on is that particular aspect of trait mining which relates to the germplasm banks and the gene bank collections of the CG. The CG holds 11 gene banks globally. They're the largest and most frequently accessed network of public gene banks globally. And we hold just over 750,000 accessions of many crop species, which are essentially the staples of the CIMMYT diet. And this is around 10% of the total ex situ collection held globally. 94% of all of the germplasm distributed globally in alignment with the International Treaty on Plant Genetic Resources for Food and Agriculture is distributed by the CGIAR. And of that, around 70% comes from CIMMYT annually. So wheat and maize going globally to a series of partners. Now, the question when we look at germplasm banks, this is the beauty of the diversity that we see. Now, that taken, most breeders would not touch this germplasm with a barge pole. Yeah, this takes you back years in terms of your population improvement, challenges of adaptation, other linkage drag. Not all diversity is good. Breeders have done a fantastic job of getting rid of the things that are deleterious, but there are some things that haven't passed that bottleneck. And the question is, how do we move forward and make the evolution and this broad diversity that we see work for us? So a few years ago, we redeveloped a concept which we now call environmental GWAS. And now we all know what GWAS is in a broad context. And here, essentially, we change the response variable. So with a standard GWAS, you have a genotype, you have phenotype data that you collect, you run that through a series of analyses to identify associations between SNPs and traits of interest. What we do here is essentially switch out to this phenotypic variable and use a different kind of data set. Data sets more relevant to the collection environments that landraces come from, that these accessions come from, to identify associations between SNPs and variables of interest. So what do I mean by these variables? Here we extend and expand the characterization information. So in the maize collection, we have around 17,500 accessions that are collected from sites, and we know those geolocation points. We know where these materials have come from. Using the geolocation data, we are able, through the use of GIS tools, to extract short, medium, and long-term climate variables for those collection sites. Information like temperature, maximum temperatures, precipitation. And we're able to right-size this information so we capture the growing season in which the crop would have been cultivated, and other important metrics like grain and flowering time. Yeah, those periods which are most impacted by heat and drought, at least in maize. Now, we piloted some studies feeding on the back of another GWAS panel that we had, which we had actually phenotyped in the field. This panel was derived from land races, and about 2,700 of the accessions in that panel had collection site data. We took that data and we started to run it alongside in the analyses with the genomic data that we had. And this is one of the early figures that we had using precipitation. And as you can see, you can get very, very clear signal in terms of alleles associated with variation in precipitation in the collection site environments. This one happened to sit right on top of a heat shock transcription factor. Now, this early data, bits of it were published by Alberto Romero, a PhD student we had in Ed Buckler's lab. And the question was, how did we take this further? Was this really relevant in terms of something that was a target that was potentially breedable? So working with Jeff Rossabara, we've been crunching this data through further. So here you can see Manhattan plots looking at temperature, precipitation, isothermality, so the difference in daily fluctuations in temperature versus the standard oscillations you'd see annually, frequency of wet days during the growing season. And in all of them, yeah, we can get some really nice signal. When we look at the top 1,000 environmental GWAS hits that we get here, and we start to look at the phenotypic response, this is looking at yield. Yes, we see a difference when we look at SNPs that are selected under or that come from that particular analyses versus random SNPs. And when we look at yield here under low precipitation, yes, the candidate alleles that we find under low precipitation do enhance yield under drought. So we do get signal that is biologically relevant. This is also translated into transcriptome signal. Here is a candidate gene that was identified. And yes, when we look in transcription data, we do get differential regulation of that gene. So great, we can find these things. When we are now considering moving from QTLs to something that's meaningful, we need elite-based donors. Yeah, no breeder is going to put a landrace into their population improvement program. With major genes and QTLs, forward breeding is an approach that's well established. And we can potentially use gene editing initially in validation of the predicted effects of alleles, and in the future in product development as that technology is more broadly accepted and deployed in our target countries. For polygenic traits, it's a little bit of a different category. And in maize, most things are polygenic, and we need different deployment strategies. And here we start to look at genomic selection that is informed by some of the outputs of environmental GWAS. So just here, don't look at the broad scale of data. What I want to show you here is essentially the outputs of breeding products using some of these technologies. In the white here, we see a series of experimental lines. These are new donors that we've been developing for drought. This material here in the yellow is the genetic check. This is essentially the elite recurrent parent that has been used. And these are two commercial lines that we use as checks. So here we're looking at treatments under drought and treatments under optimal conditions. Yeah, the last thing we want to do is improve drought tolerance, but lower optimal yield. And as you see here, we do get a boost in all of these donors. So we are able to move polygenic variation forwards. It's not impossible. It's not simple, but it's doable. So here now we start to use environmental prediction. So we've all heard of genomic selection. Here we use genomic prediction. We're able to predict the accession and environment, the environment and accession came from with very high accuracy. And we're starting now to use this kind of paradigm to predict the potential of a landrace. What is the genetic potential of a landrace within a particular environment? These predictions, this is a nice slide that came out from Jeff Rosabara recently. We're able to use this information now to predict a genotype of an environment that may not have been sampled. So this is work looking at the landrace collection from Mexico and trying to say here, in this particular environment, what would the ideal genotype be? And then go back to the existing data and validate that. And this is proving pretty useful at the moment. So how do we use this knowledge in terms of pre-breeding? We use the environmental GWAS to identify novel variation, novel adaptive variation of breeding relevance. Sometimes we find large effect QTLs, they're very useful to move forwards. We're able, through comparison with the existing material in terms of the elite germplasm, to identify those alleles which are novel to the elite germplasm. The last thing we want to do in a breeding purpose is repackage something that's already been captured by breeders effectively. The prediction we use to get a rank order of those land races which may have higher value under a particular environmental stress. And we use this information together to identify those accessions which have the highest novel value for breeding. And these form the breeding starts for the development of new elite base donors. And with that, at 45 seconds over time, thank you very much for your interest, and I hand it back to Mark.